Hello everyone. So it's 12 p.m. which is the prime time for the plants to bask in the sun. For us, let's bask in botany wisdom. A very good afternoon to everyone. So I am Bushra, a botany faculty, and today I'm going to discuss the questions which are mistaken by lot many students in this ongoing series of AIATS plus phase 2 which was held on 2nd of February 2025. So in this session, I'm basically going to discuss questions from these topics. Okay, the first two chapters are from class 12. First one is sexual reproduction in flowering plants and second one is organism population. These two chapters are very important from NEET point of view as lot many questions are asked from this chapter as well as from this chapter particularly from population interaction. And the next four chapters are from class 11th, plant kingdom, cell the unit of life, photosynthesis and morphology of flowering plants. So these are the chapters on which other questions I am going to discuss in this session. Now without wasting any time, let's start with the very first question, which is question number 96, and it says match the placental types column 1 which was given in column 1 with their examples in column 2. So what you understand by placentation? It is basically the arrangement of ovules in the ovary. So if we talk about the gynosium part in the female reproductive part of flower, a very good afternoon to everyone. Please keep joining, okay? Now let's start with the placentation type. So here I am talking about gynosium, which consists of three parts: a stigma, style and ovary, right? Now let's make it for you. For example, this is stigma, this is style and this basal part is ovary. So where these ovules are basically arranged, they are arranged in the ovary part, in this region, right? And placenta is the surface, or we can say the flattened cushioned surface on which ovules are born on it. Now let's start discussing the types of placentation which we could see in the plant. Before that, I would like to tell you that if gynosium contain carpels, if more than one carpel is present and if they are free, when carpels are free, then they are known as apocarpus. Tell me the example of apocarpus, class. Okay, when carpels are fused, then it is known as syncarpus. For syncarpus, the example is tomato. For apocarpus, lotus. बाकी example आप मुझे बताएंगे. Now, let's proceed in the placentation type. The first one is, let me erase it for the space. I hope you people have noted down. Thank you. Now, let's move. The first one is marginal type. Why it is known as marginal type? Because the placenta is formed at the margin of the ovary and from the ridge along with the ventral suture of the ovary where ovules are formed. So if you could see the diagram, so this is the placenta and these are the ovules which will later form the seeds. Okay, so this is marginal placentation and it is found in P, or we can say the most member of Fabaceae family. यहाँ पे P जैसा दिख रहा है ना, so from this we have to remember that marginal placentation is found in P. Now next one is axial placentation. So in this, placenta is in axial position and we can see that how ovules are present in the different locule. These are locules. These are ovules which are present in the locules in multilocular ovary. And this type of placentation is found in the members of Solanaceae and Liliaceae. So this is how we can relate the concept from one to another one, right? Very good, Aman, very good. You remember all the example, very good. Now next, so example for axial placentation is tomato, China rose. Right? Now let's move ahead with the next placentation type, which is parietal. Here you can see that how ovules are attached to the inner ovary wall. This is inner ovary wall, and ovules are attached to it. Right? तो यहाँ पे placenta है जिससे ovules attached हैं. And this type of placentation is observed in brassica, that means in mustard, and in argemone. So these are the example. In them, false septum is also observed. So in NEET exam, sometimes question have been asked previously that false septum is observed in which type of placentation? So the answer is parietal. Okay. Now, next one is free central placentation. Free central placentation. Why it is so? Because ovules are present on the free central axis. So this is the placenta, right, on which ovules are present. And it is present at the center, as you can see in the diagram, and no septa is observed. कहाँ बनेगा septa? जगह ही नहीं है. So in this, no septa is observed, right? And इसके examples हैं dianthus and primrose, right? Be particular about examples. Now the last one is basal placentation. Why it is known as basal placentation? Because placenta is present at the base of the ovary and ovule is attached to it. It is found in sunflower and in marigold, or we can say particularly in the members of Aestivation family. So this is how basically we connect the concepts that I have told you, the names of the family through which all these placentation plus type of placentation are belonging. So this is the complete, or we can say the difference between different types of placentation. Now let's get back to the question. The first one is basal. So basal placentation is found in sunflower. So from this only I can hit the correct answer, which is given in option 1. So this is how we strategize our question to choose the correct answer. Okay. Now next one is exile. Exile placentation is found in China rose. China rose is the member of Malvaceae family. Along with the Malvaceae family, in which two families exile placentation is observed? Please comment down your answers. Okay. Now next one is parietal placentation, which is found in mustard, given in 1. Last one is free central placentation, which is found in dianthus along with argemone. So from this I can hit the correct answer, that is option number 1. Okay. Now let's move ahead with the next question, that is 100, which says select the correct statements with respect to plasma membrane and choose the option accordingly. So here certain statements are given for plasma membrane. Now let's see that actually how plasma membrane looks. So this is the model of plasma membrane which is given by Singer and Nicholson. Right, so before that, how we actually reach to this model, at times, when times goes on and our microscopy techniques get improved, so with the evidence of that, this structure is able to find. Now, how this, the main composition of plasma membrane is lipids and proteins. Right, and lipids they are mainly composed of phospholipids. If we talk about phospholipids, they are arranged in bilayer form. how they are arranged? Let's see. This is the head, right? This is the tail. This head is polar. That means it is towards the aqueous region of the cell. And this is the tail, which is hydrophobic. That means it is away from the aqueous region of the cell. If it is arranged in a bilayer, then again, second layer, which is arranged in a reverse manner, this is tail and this is head, which is polar in nature. Right? As you can see here in the diagram that how they are arranged. Right? So these are basically lipids, phospholipids. Now, in the cell membrane, you can see that there are lots of structure which are protruding outside. This is the integral protein which is buried in the cell membrane or plasma membrane. These are integral proteins. And if we could see that these are proteins which are present on the periphery. These are peripheral proteins which are present on the surface of plasma membrane. Right? Along with that, they have the attachment of phospholipids. Now, in the plasma membrane structure, you can see these yellow structures which are present somewhere in between the phospholipids layer. These are cholesterol. Okay? These are cholesterol. Now, these peripheral and integral proteins which are found in the plasma membrane are basically classified on the basis of their ease of extraction. So this is the whole idea of a cell membrane or plasma membrane. In which cell it is basically evident, it is first extracted from the membrane of RBC or erythrocytes in humans, in which if we see that 40% is lipids and 52% is protein. If we talk about the composition of plasma membrane of RBC, then it composed of 40% of lipids and 52% of proteins. Please remember these terms as this has been asked in the NEET exam. 40 for lipids, 52 for proteins. Okay? Yeah, very good, Sagnik Datta. Now, if we talk about the functions, that what function this plasma membrane performs, it performs several functions. For example, secretion, endocytosis, signaling, exocytosis, etc. But the main role of plasma membrane is transport. If we talk about the main role for plasma membrane, it is transport. What is the nature of plasma membrane? It is selectively permeable. That means it is not permeable for each and every molecule. It is selective for its molecule, that which molecule can pass through it. Now, this transport can occur in three different means. The first one is passive. Second is facilitated. Third one is active. I am writing here for you. Passive, facilitated and active. What do you understand by passive? In which we do not require any energy. That means the substance can pass through one area to another area via membrane with its concentration gradient. That means from higher concentration to the lower concentration without the use of energy. If we talk about the facilitated transport, for this, the substance will pass through the plasma membrane by the use of certain substance, or it is get facilitated by certain substances. For example, water will pass through the membrane via facilitated transport, which require porins. to pass through it. Last one is active transport, in which it requires energy in the form of ATP, and its example is sodium-potassium pump. Please remember that sodium-potassium pump is the example of active transport which requires energy to pass through the membrane. Okay, so this is all about the cell membrane structure and its function. Now let's get back to the question where statements for plasma membrane is given. The first one is tail of lipid is polar, whereas head is non-polar. This is incorrect because in contrary, the tail of lipids is non-polar where head is polar. That's why this statement is incorrect. I can rule out option how, that in options which contain A will directly get ruled out. That means option 1 and option 3. Now let's move ahead with the next statement, which says integral proteins are totally or partially buried in the membrane. So yes, it is absolutely true. As you have seen that these integral proteins, these big structures are integral proteins which are completely buried, or some they are partially buried. Okay, so this statement is true. So here we have to choose the statement which contain B. So 2 and 4 are still there. Now next statement, which says in human the membrane of RBC has approximately 40% protein and 52% lipid. So it is incorrect statement. Why? Because 40% of lipids are present and 52% of protein is present. So this statement is incorrect. Please don't confuse in these two numbers. Remember like that lipids start from L, low number 40, like that. Okay. So from this I can rule out option 2 as C is incorrect statement, and question is asking about correct statements. Now statement D, which says proteins move laterally within overall bilayer of lipid. So yes, this is absolutely true. One thing which I left in the explaining concept, that the model which was given by Singer and Nicholson, they are the person who gave fluid mosaic model of plasma membrane. Right? What they have said that quasi-nature of lipids, that means the semi-fluid nature of lipids, enable the movement of proteins between the bilayer of lipids. This bilayer of lipids के बीच में they enable the movement of proteins, and this is known as fluidity, and they termed it as fluidity. Remember that protein move laterally within the overall bilayer of lipids. This is the idea for a fluid mosaic model given by Singer and Nicholson. So this statement is correct. So among the 4 statements, 2 statements are correct. Those are B and D, which is correctly depicted in option 4. Okay, now let's move ahead with the next question, that is 106. And it is from the chapter Photosynthesis in higher plants. Now let's see the question. Again, statement type question. So you can see that this is the pattern of NEET till now, that they ask the question based like in statements. So it may consume your time, but they are easy question. You just know simple trick that how to solve all these questions, how to rule out the options likewise. Okay. Now let's see ATP synthase ka role during chemiosmosis. So this is the beautiful diagram from the NCERT itself. This is the thylakoid membrane. We are going to talk about chemiosmosis in chloroplast. This is the stroma region. Okay, now let's start that how basically this works. So first of all, I would like to start from that who has explained this thing. It was first explained by P. Michel. Right? This chemiosmosis hypothesis in chloroplast. So he basically explained that how ATP got synthesized during chemiosmosis, or we can see that how protons get accumulated and help in the formation of ATP. So there are different ways in which protons get accumulated, or we can say gradient is formed across the membrane so that ATP can synthesized. Okay, now let's start that how they proceed. First one is that how proton gradient is formed. First one is photolysis of water. That means splitting of water. If you remember the Z scheme from the photosynthesis chapter, where splitting of water takes place near the PS2, that means which is facilitating the splitting of water, that is PS2. Here H2O, you can see that H2O getting splitted into half O2 and H plus. This H plus will contribute in the lumen side of the chloroplast where they get accumulated. So this is one of the way. Now next, this electron is getting passed through the plastoquinone. Here this electron is getting passed through the plastoquinone. Meanwhile, this plastoquinone is transferring the protons from the stroma region to the luminal region. This is also increasing the concentration of H plus or protons in the luminal side, right? So the next one is that how transfer of protons. Transfer of protons from stroma to lumen via this transport or this carrier molecule. Right now, the third way, due to which the concentration of proton is getting increased. एक साइड का concentration increase करना है, the either way is to decrease the concentration of other side. So how it is actually occurring? There is one enzyme known as NAD reductase. There is one enzyme which is known as NAD reductase. NADP reductase, I'm so sorry. What it does, it basically accepts the protons from the stroma side and it get added with the NADP plus and it get formed into NADPH, which will act further as a assimilatory power in the photosynthesis, right? So what it is actually doing, it is decreasing the number of protons in the stroma side, due to which a gradient is formed in which we can see that the number of protons in the stroma region is quite less as compared to the protons which are found in the luminal region, which will lead to further decrease in pH. I hope you know this concept that increase in hydrogen ions inside the cell will decrease the pH, right? Okay, now अब यहाँ पे हाइड्रोजन इंक्रीज हो गया है ल्यूमिनल साइड में, राइट? From breaking this proton gradient which is formed in the across the membrane, how this proton gradient will further get break to form the ATP, which is facilitated by the enzyme known as ATP synthase. This is ATP synthase enzyme, which consists of two parts. First one is CF0, which is embedded in the thylakoid membrane, right? And it forms the channel for the transport of proton through it. CF0, please remember that CF0 is embedded in thylakoid membrane, and the next part is CF1, which is protruding on the surface of outer membrane of thylakoid, right? Protruding outside. Please remember these things as this has been asked in the previous year NEET questions. Now, how it will get form ATP? From here, protons will move through the CF0 particle, right? And then further from in the CF1, they will change the conformation of CF1, which will lead to the formation of ATP by ADP and phosphate group, right? So this is how ATP gets synthesized during the chemiosmosis. One more thing, remember that NADP reductase enzyme is present towards the outer side of the outer membrane of thylakoid membrane. Outer thylakoid membrane, okay? This is asked in NEET, previous year NEET exam. So this is the complete case. Now let's get back to the question. The first statement which says it pumps protons across a membrane to create a proton gradient. Here is the confusion that it create a proton gradient. ATP synthase, that means we are basically talking about this region, only this enzyme. So it is not creating the proton gradient, it is breaking the proton gradient to form the ATP, right? So this statement is incorrect. Now, next statement which says it includes a channel that allows diffusion of proton back to stroma across the membrane. So yes, this is also correct. Sorry. So yes, this is also correct, which is flowing back the proton back to the stroma. It is CF0 particle in which the channel, transmembrane channel is present. So this statement is correct. Now, next one is catalyzes formation of ATP. Obviously, as the name itself suggests, ATP synthase, that means synthesis of ATP, right? Now, the fourth statement which says it helps in splitting a water molecule on inner side of the membrane, which is not facilitated by ATP synthase. On the contrary, it is facilitated by PS2. Right? So this statement is incorrect. So among the four statements, two statements are correct. Those are B and C, which is correctly depicted in option 2. Okay? Now let's move ahead in the next question, which is 110, which says scutellum is. So this question is from sexual reproduction in flowering plants. Now let's see. This is the beautiful diagram from the NCERT itself. This is the embryo of dicot, and this is the TS of embryo of monocot. Now let's see what is the difference in between. If we talk about the embryonal axis, let's understand this thing likewise. This is embryonal axis. On the upper side of the embryonal axis, epicotyle is present, where it will lead to the formation of coleoptile, right? Or we can say, if we talk about the dicot, here it is giving to oops, I'm so sorry. Yeah, epicotyle, which is giving rise to shoot apex. Now, if we talk about below the embryonal axis, it is hypocotyle is present. It will give rise to shoot apex. This is the idea, or we can say this is the way we can remember this. Remember, this is embryonal axis, which I had made. This is only the purpose for explanation. Please don't consider it as a real diagram of the dicot embryo. So above the epicotyle, a shoot will form the shoot apex. Above the embryonal axis, epicotyle is present and shoot apex will be formed, and then hypocotyle, hypocotyle root apex is formed. Yeah, it's root, not shoot. I'm so sorry. Now, if we see this diagram, so this region is labeled as hypocotyle. These two lines are labeling cotyledons. As the name is dicot, that means it will have two cotyledons. Right? So this is radical, or we can say root apex, and it is enclosed in a root cap. So this is all about the dicot embryo. Now if we talk about the monocot, again the embryonal axis above which epicotyle is present, and on that coleoptile is there, which will further get enclosed, which encloses the shoot apex. Coleoptile encloses the shoot apex. What is coleoptile? It is the holofolia structure. This is the holofolia structure. And if we talk about towards the downward side, that is hypocotyle, it encloses the collier hyza, which further enclosed root apex. What is collier hyza? This is undifferentiated sheath of mass which encloses the root apex in monocot embryo. And in monocot there is already one cotyledon, and it is known as scutellum, which is depicted here. This region, this which we can see here, is shoot apex. Right? This is root cap, which is enclosed in the collier hyza. And above this is radicle. This is how the complete structure of monocot embryo looks like. If we see the question, it is asking about the scutellum. The first option is remnant of nucellus. If we talk about remnants of nucellus while the development of embryo, then it is known as perisperm. And the seed in which perisperm are found are known as perispermic seed, for example, beet or pepper. Like, so this is incorrect option. Now let's see the next one, which says a sheath that encloses plumule. So no, this is not the case. The sheath which encloses plumule is coleoptile, which I have told you in the diagram, which encloses the shoot apex in the monocot is actually known as coleoptile. Third one is single large shield-shaped cotyledon in monocot seeds. So yes, this is absolutely true. Scutellum is the single large shield-shaped cotyledon. And what happened to the next cotyledon? If we see the diagram, this region, it is actually not written in the NCERT but given in the diagram, which is the epiblast. It is the remains of second cotyledon found in the monocot. Okay. Now, option 4, which says endosperm of monocot seed. So no, this is also incorrect. So for this question, the correct answer is option 3. Now let's move ahead with the next question, which is 122, which says, below is the diagrammatic representation of response of organisms against temperature. Find out the correct match. So this is the diagram. The graph is given. This question is from Organism and Population. It is showing that how organism is reacting towards the different abiotic factors in the environment and how they are actually surviving. Now let's see. Let's understand this thing from this diagram. So conformers, regulators and partial regulators are basically the type of organism which are classified on the basis that how they actually respond to the abiotic factors. So first one is conformers. Conformers. So these are the organisms which could not able to maintain temperature if the temperature get changes in the environment. Right, say they do not have the ability to maintain their internal environment with respect to the external environment. So these are conformers, for example, plants. If we talk about next organism, type of organism, which is regulators. Regulators are the organism which can maintain their internal temperature, or we can say it can maintain their osmotic pressure or potential inside their body. Those are regulators. That means they are able to regulate their body temperature and conditions. For example, all birds and mammals. You have seen that we have the ability to survive in any condition. If we are present in the Sahara Desert, then also we are surviving. If we are present in the Antarctica area or Arctic area, there also humans are surviving. That means we have the capability to maintain our temperature. Irrespective of the condition, what is the condition of outside in the environment? So we are regulators. We have the ability to maintain our temperature, and this ability is known as homeostasis. So how this is actually, these organisms are able to maintain their temperature and osmotic pressure regulation is via thermoregulation and osmoregulation. So these are the way from which regulators maintain their body temperature. Right, so if we come back again to the conformers where organisms are not able to maintain their temperature with respect to the external environment. So we could see that there are no smaller organisms which we could find in the polar region. So why it is so? If we compare the surface area to volume ratio in the smaller area, then they have large amount. Like if we calculate the surface area for the smaller organism, it's much way higher than its volume, due to which it will lead to the loss of heat from his or its body excessively, right? And if in comparison to the larger body, the surface area to the volume is less. In comparison to their volume, the surface area is less. That's why they are able to maintain it. So conformers which are not able to maintain their they are not able to maintain their body temperature from the outside environment, actually larger in size in the polar region. So that is the region, that conformers which are found in the polar region. The third one which is mentioned here is partial regulators. Partial regulators, that means these are the conformer animals only, organisms only, but up to certain limit they are able to regulate their temperature, but after that, after a while they can again become the conformer, right? So this is about the two types, or we can say two types of response which the organism exhibit. Apart from these two, one more is known, which is migration. Migration, in simple terms, that organism move from unhospitable condition to the hospitable condition, and once the condition become favorable, they come back. For example, there are certain birds which come in Rajasthan Kevladev National Park in winters. Please remember the name of this national park, as it has been asked in the NEET exam previously. Okay, so this is one of the way. Next way is suspension. So what they does in this, they basically suspend their metabolic activity during the unfavorable condition. Yes, very good, Sagnik Datta. Shreya and hummingbirds, these are what? Please mention that. Shreya and hummingbirds क्या होते हैं? बताइए. Now, the next one is suspension. So, type of suspension organism show. So, if we talk about fungi, how they suspend their metabolic activity by forming spores. So, spore formation is one way. Next is hibernation. Hibernation is related to escape from the cold environment, right? Now, next one is aestivation. It is one of the way through which organism can escape from summer-related problems. And the fourth way is diapause. It is generally observed in the zooplanktons. They suspend the growth of the development, and this condition is known as diapause. Remember that hibernation is related to the winter and aestivation is related to escaping the summertime. For example, snails. For hibernation, example is bear. Right? So these are the responses of an organism which will be exhibited by them in abiotic stress. Now, let's get back to the question where it is asking about A and B. So what is A? It is conformer and B is regulator. So हमारे regulators कौन थे? Birds. Birds in given in option 1 only. So from this only I can hit the correct answer, and A है हमारे conformers, which are plants which are not able to maintain their temperature. So the correct answer is option 1. A student is asking, isn't hummingbird a bird? Shouldn't it be a regulator? Hummingbird, it is not actually that all birds and all organisms are able to maintain the temperature. There are obviously certain exceptions in the body, in the nature, which we can see. Okay. Now, let's move ahead in the next question, that is 1 to 7, which says match the following columns and select the correct option with respect to aestivation in corolla. So it is asking about the aestivation in corolla. What is aestivation? This is the arrangement of calyx or corolla in the flower whorl. Now, if we see the first one, which is known as valvate aestivation. Here we can see that the sepal or petals are arranged in a very defined manner where they are not touching with each other in any way, and it can be observed in Calotropis. It is also observed in the members of Solanaceae and Liliaceae. Solanaceae and Liliaceae. Members of Solanaceae and Liliaceae. So, see, this is how we learn the concept, or we join the concept, either way. Next one is twisted aestivation, where we can see that sepal or petals are actually overlapping each other in a very defined manner. Like this is first one, next one is overlapping the first previous petal likewise, and it is observed in China rose. Right? In China rose, which is the member of Malvaceae family, a very beautiful aestivation is observed. Two different type of aestivation. First is twisted, and in calyx, if we talk about the aestivation of calyx, we observe valvate type of aestivation in China rose. Please remember that for the members of Malvaceae. Let me put it in a box for you people so that you can remember this. It is particular for the members of Malvaceae. Now, next one is imbricate, where we can see that how petals are not arranged in a defined order. They are arranged in very ill-mannered way, not in a particular defined order, and it is observed, this type of aestivation is observed in Cassia and Gulmohar. Now last one, which is very interesting aestivation, one of my favorite. It is known as vexillary aestivation. Very beautiful aestivation. It is observed in the members of Fabaceae. It is also known as papilionaceous aestivation because it is particularly found in the members of Fabaceae, which is earlier known as Papiliondae, right? And if we talk about this aestivation, the first petal, which is the largest one, is known as standard, which is also known as vexillum or carina. These terms I am writing because again it is asked in the NEET exam. Now these two petals, which are placed next to the standard, they are enclosed by the standard petal, they are known as wings, right? And here you can see that these two petals, which are fused with each other, the smallest one, they are known as keel. Please remember this labeling of the vexillary aestivation, as it is very important for exam point of view, and it is observed in the members of Fabaceae, for example in P. So this is how we remember different type of aestivation in a flower. Yes, Sagnik Datta, very good. The largest one is standard, which overlaps the two lateral petals. Yes, the wings are lateral petals, and in turn they overlap the smallest anterior petal. Very good. Now let's come back to the question. Here we have to match them. The first one is, what type of aestivation is observing? In the first one, it is imbricate, right? Let's mark them first. Next one is valvate. C is vexillary. And the fourth one is twisted. So first one is imbricate. Imbricate aestivation is observed in Cassia. So A matches with 3. So from this I can rule out option 1 and option 3, as they are claiming that A matches with 2. Now next one is valvate. Valvate aestivation is observed in Calotropis. So B matches with 1, which is correctly depicted in option 4. Right. So from this only I can hit the correct answer. But let's move ahead. Next one is vexillary, which I have told you they are particularly found in the members of Fabaceae, and the member of Fabaceae is bean. So C matches with 4. Now the D one is twisted. It is found in cotton, right? Cotton is also the member of Malvaceae, and the important feature which I have told you about Malvaceae, please remember that about the difference of aestivation in calyx and corolla, right? So from this I can hit the correct answer, which is option 4. So this is how we basically hit the correct answer in these bulky questions. Now let's move ahead with the next question, that is 134, which is assertion and reason. Assertion reason dekhte hi we are like, oh my god, how we are going to solve this question, but they are actually very easy. Now let's start. Assertion, which says embryo development precedes endosperm development. First of all, we have to look whether the assertion statement is true or not. It is saying that embryo development precedes, pehle hoti hai, then endosperm development. So no, this is not the case, because endosperm will precede the embryo development, because for the formation of embryo, the food supply will be get from endosperm. So this is not the case. This statement is itself wrong. Okay? For example, अगर precede का मतलब नहीं समझ में आता तो ऐसे याद रखो that 2 precedes 3. 2 पहले आता है 3 से, likewise. Okay? Now, so from this, I can easily rule out first two statements, or I can say all the three statements if I know that first statement is incorrect, because in all the three options disclaiming that assertion is true, but this is not the case. Now, let's see the next statement, which is reason, which says early stages of embryogeny are different in both monocots and dicots through seeds. Those seeds are similar. So no, this is not the case. If we see the early stage of embryogeny, that means the formation of embryo in dicot and monocot, then we can see that they have similar stages. They are same. But the seeds are different, as I have shown you in the previous question, that how monocot and dicot seeds develop differently. Right? So this statement is also incorrect. So from this I can conclude that both assertion and reason are false statement. So this is how basically we solve the question. But if we have to make the assertion and reason correct, for example, let's see in this question only that endosperm precedes the embryo development. If we correct it, okay, and if the reason is same, that early stage of embryogeny are same in both monocot and dicot seeds, then also the answer will be not there. Because if we put because in them, after the first statement, after the assertion, if we put because and read the statement, then we will see that it is not making any sense. These two are independent statements. So from that statement, I can see that it is true, that both A and R are true, but reason is not the correct explanation of assertion, right? So this is the way how you solve the assertion and reason question in the exam. Okay? Sanjay Ghatak, yes, very good. Sagnik Datta is giving very correct answer, very good. So this is all about this session. So these are the questions which are basically mistaken in this topic. So I hope this is the lunch time. Have your lunch, make up energy and get back to the zoology session at 2 p.m. Thank you so much and have a great day ahead.